Hello and welcome to this short video presentation, Aerosol Testing a Cuvette Flow Cell-Based Cell Sorter, presented by the International Society for the Advancement of Cytometry and CytoU. In this short video presentation, a step-by-step breakdown of aerosol containment testing on a Becton-Dickinson Vax Aurea cell sorter will be described, following the Cyclex-D Dragon Green bead method described by Stephen Perfetto, Kevin Holmes, and the ISAC Biosafety Committee in their 2018 Cytometry Part A publication. Remember, prior to performing any work with biohazardous material, a thorough risk assessment should be performed to identify the hazards, personnel, procedures, risks, and mitigation strategies as they apply to your specific situation. Consider discussing this procedure with your institution's biosafety officer and internal biosafety committee. And remember, it doesn't matter what I do, what a manufacturer does, or what anyone else does. It only matters what you do in your institution with your instrument and your users. My name is Jellison, Director of Flow Cytometry at the University of Connecticut School of Medicine, and I'll be your narrator for today's presentation. The objective of today's presentation is to demonstrate the steps required to perform an aerosol containment test on a commonly used Cuvette flow cell-based cell sorter. We will go over the materials required, instrument setup, how to synthesize a clog, testing in both containment mode and failure mode, and finally result analysis. In addition to your cell sorter, here is a list of the additional materials you will need to perform this testing. A vacuum pump, a rotometer capable of reading 20 liters per minute, 1 micron Dragon Green beads, Cyclex-D cassettes, microscope slides, and a fluorescence microscope capable of seeing those Dragon Green beads. To begin, set up your instrument and start the stream with the highest pressure and nozzle that will be used for cell sorting. Turn on and verify that the aerosol management option is set to the correct evacuation rate. Prepare the Dragon Green beads by diluting them 1 to 100 in 1 to 2 milliliters of saline solution. Load the Dragon Green beads onto the cytometer and try to attain an event rate of approximately 50,000 events per second. Open the sort chamber drawer and place a split piece of tubing over the end of the center waste stream collection notch. This tube will deflect the stream upon opening the sort chamber drawer, allowing for the generation of aerosols which contain the 1 micron Dragon Green beads. These will be collected by the Cyclex-D cassette. Close the sort chamber door. Remove all collection devices and device adapters from the collection chamber. Place a paper towel or small plastic container beneath the sort drawer to collect the upcoming mess. Prepare the Cyclex-D cassette by removing the protective film and insert the cassette on the end of the vacuum line. Double-check that the aerosol containment module is running as intended. Place the Cyclex-D directly in front of the opening of the sort chamber, where the collection device adapter would normally hook in. Open the sort chamber drawer. Turn on the vacuum pump. Check that it is drawing 20 liters per minute and sample for 10 minutes. Upon completion, turn off the vacuum pump and remove the cassette. This Cyclex-D sample will test if aerosols have been contained during normal operation. No Dragon Green beads should be detected on this Cyclex-D cassette. Aerosol testing in failure mode. Turn off the aerosol containment module. Load a new Cyclex-D cassette on the end of the vacuum tube and place it directly in front of the opening of the sort chamber, where the collection device adapter would normally hook in. If it was not left open, open the sort chamber drawer. Turn on the vacuum pump. Check that it is drawing 20 liters per minute and sample for an additional 2 minutes. Upon completion, close the sort chamber drawer, turn off the vacuum pump, and remove the cassette. This sample is a positive control. 100 or more Dragon Green beads should be collected on this Cyclex-D cassette. Analyzing your results. Crack open the Cyclex-D cassette to reveal the glass cover slip containing adhesive and aerosol sample. Carefully remove the cover slip from the device without touching the adhesive, and mount the cover slip, adhesive side down, directly onto a microscope slide. Here, our model Jeff is using a gridded microscope slide to aid him in focus and allow for easier scanning. Examine the slide from the failure mode cassette. You should see at least 100 beads. By contrast, the containment mode cassette should contain no beads. Shown here are two examples from one field of view for each sampling situation. In summary, no dragon green beads should be found on the containment mode slide. The positive control failure mode should have collected at least 100 dragon green beads. In addition to containment mode and failure mode, you may wish to test your SOP to determine when it is safe to open the chamber door after a nozzle clog. For instance, a common standard operating procedure may be that, after a nozzle clog, one would wait for a period of time to evacuate aerosols from the collection chamber, then open the sort drawer and wait additional time to evacuate the sort chamber. In this example, Cyclics D sampling should be performed for 2 to 5 minutes after each evacuation step. I would like to thank Jeff Lyon, the primary videographer and model for this presentation, as well as Stephen Perfetto, Kevin Holmes, and the ISAC Biosafety Committee. The original publication of this procedure can be found in Cytometry Part A or on the ISAC Biosafety website, where you can also find other great flow cytometry-related biosafety information. We have reached the end of this presentation. Thank you for tuning in. Be sure to check out Parts 2 and 3. And for more great cytometry-related educational content, please visit us on the web at cytou.org. On behalf of the International Society for the Advancement of Cytometry, thanks for joining us.